All right, let's get started for this afternoon. Hello, everyone, and welcome to the CHIP Landmark Ideas series. It gives me great pleasure to introduce Atul Butte, who is going to talk to us today about precisely practicing medicine from 700 trillion points of data. Atul is, if to many of you, needs no introduction, but I will briefly introduce him. He, first of all, I've known Atul since he was a resident, and then an endocrine fellow, and then a faculty member here in the computational health informatics program at Boston Children's Hospital. He has gone on to great things at the University of California, where he is now the Priscilla Chan and Mark Zuckerberg Distinguished Professor and Inaugural Director of the Bakar Computational Health Sciences Institute at the University of California, San Francisco. He is also the Chief Data Scientist for the entire University of California system. And this laboratory of his is going to form the basis, I believe, of his talk today. I want to do a couple of housekeeping things, as they say, and then I'm going to turn it over to Atul. So you can put your questions into the Zoom Q&A box as we go. The schedule is that we'll do about 50 minutes of Atul, and then we will go into a Q&A. I'll ask a few questions, and then we'll go to questions from all of you. The brief background on CHIP: we were founded in 1994 as a multidisciplinary applied research and education program at Boston Children's Hospital. We're at www.chip.org. The Landmark Ideas series offers points of view and talks from unique thought leaders across healthcare. Previous speakers have included Nick Christakis, Ricky Bloomfield, a health lead at Apple, David Clark, an inventor of TCP/IP, therefore an inventor of the internet, and others. You can learn more about this and sign up to learn more under chip.org events. If you want to tweet, we have for you a convenient hashtag. Atul is always looking for followers. And you can also follow us at Boston Chip. And again, the questions, if they're queued up in advance of Atul's finishing up, it helps us get right into the questions with no pause. And with that all said, I'm going to turn it over to today's Landmark Ideas speaker, Atul Butte.
Great. Thank you, Ken. Let me just start the screen sharing here. All right, thumbs up. You're seeing that? I'm guessing yes. All right.
Perfect.
All right, so thanks, Ken, for having me. It's great to virtually be back at Boston. Obviously, 10 years of my life there. And I can already see of the 210 participants, many names of people I remember and know. So it's great to see you all there. Obviously, first, I'm a medical doctor. Most of you are medical folks, so I got to start with my disclosures. I have just a few to disclose. Actually, probably more than a few. I'm obviously involved with a lot of different companies, but suffice it to say at the top left, I've started a couple companies. I consult for a whole bunch of companies. You might not want to believe another word I say over the next 50 minutes. I wouldn't blame you. But I'm also proud of the right side of the slide, which are all the companies started by my students and some by postdocs. I do often say in my lab that if you want to change the world, you can't just keep writing papers about it, right? You actually have to do something and take it out of academia. So if you've invented something or discovered something, it's up to you to license it out. And if it doesn't get licensed, then it's up to you to really start the company and make sure patients, families, and physicians and nurses all benefit from what we are doing in the academic labs. So a lot of docs use this as a slide of shame. This is my slide of significance. And with that, I'll get started. So obviously we're in this data-rich world. Pick up any magazine, you'll see the covers. You know what I'm talking about, whether it's Fortune or Science or Harvard Business Review, or of course, our scientific journals. Nature, and you see Time Magazine on the right there. Actually, The Economist at the bottom keeps covering it again and again. And my favorite one is on the bottom right there, which is kind of interesting, right? They say data now is the most valuable resource, and the analogy there is it's as valuable, let's say, as oil, right? You see oil rigs there. In fact, people use this saying, right? Data is the next oil. I hate that saying, because oil, if you think about it, if you think about a barrel of oil, either I take that barrel of oil or you take that barrel of oil. We both can't have it at the same time, right? But data, of course, I can have a copy and you can have a copy. And I might do magical things with it, and you might do differently magical things with it. And we might actually never compete, even though it's the same data, right? I'm going to give you a lot of examples of those. So the saying I think I like better is, it's not, I didn't invent the saying, but the saying goes that data is the next soil, right? Not oil, soil. So you plant ideas and data helps them grow, right? So that's the kind of concept I want to resonate with you all here today. But really to level set, because there's a lot of people from the public here, to level set a little bit, I want to just make sure we're all on the same page. Here are three phrases that are used a lot: artificial intelligence, machine learning, and deep learning. I want to make sure we all get those phrases, because obviously data now is moving into the use of that data, and the big popular ones are AI and machine learning. How do you sort out these phrases? Well, one way to think about these phrases is artificial intelligence is the top category here, so it's the broad one. AI is mimicking aspects of human intelligence by computers, so translating languages, cars driving by themselves. The machine learning is one kind of AI where you're teaching the computer using data. Now you might think about that and say, how else would you teach a computer except by giving it data? Well, in the old days, not so old days, we didn't have a lot of data, so we used to use rules. So we ask an expert, when you see this, what do you do next? What if you see that, what do you do? What do you do to take care of that patient or that condition? But instead of rules, now we've gone to using data. Because in my view, it's much better for example, using the medical area, which we will be, it's much better to see what a doctor does than ask them what they do, right? Because if you have all the orders, and you'll get to that, then you can see obviously it's much more rich than asking the doctor if they remember what they do in this case or that. But machine learning is using data to teach computers a set of rules. And under that, we have supervised and unsupervised. So supervised means that we have a right answer, right? We know if there was pneumonia or not in this test X-ray. We know if there was a fracture or not, right? And all we're trying to do is get the computer to be as good as the current state of the art is, right? So we can measure things like sensitivity, specificity, accuracy. We can give it a right or wrong answer. Unsupervised means we really don't have a right answer. We have a lot of data. Help us, computer, help us organize this data. Clustering, k-means clustering, you might have heard dendrograms, hierarchical clustering, stuff like that would be unsupervised. And then under that is deep learning, which is one kind of machine learning where the software architecture crudely resembles neurons or layers of neurons in a brain, but it's really kind of a crude resemblance there. Now, all of this stuff sounds really sophisticated, but really the idea here is to demystify as much of this as possible. Indeed, there are dummies books on all three of these subjects here, right? Literally $17 on Amazon Prime. All these are software tools that you put into your software to get this magical, seemingly magical stuff to happen, right? You download this software, it's increasingly free, you put it in, and then these things happen. Now, of course, it's changing medicine already, right? So in fact, we literally have blog posts saying AI is already changing medicine. We have Eric Topol with his best-selling book, Deep Medicine. We have some journals that are so old, they're new again, like Artificial Intelligence in Medicine. We have some fields of medicine and journals like radiology that have already come up with radiology AI, right? New journal there. And then again, I'm speaking on the medical side of things. We have JAMA and Nature Medicine, both of which had very different types of papers before, but both of which have now opened up for AI and machine learning types of papers and approaches here. And of course, we have products and services. So here are the first three that were approved by the Food and Drug Administration, the US FDA. And you can see the earliest one is January of 2017, so not that long ago. These are cloud-based deep learning tools. The one in the back there is by a company called Arteris, where for cardiac imaging, the software helps us figure out, is there something wrong with the heart in terms of the vasculature, the blood flow. Viz AI is for stroke triage. So this is, for example, an emergency department. And then IDX is one of many groups and companies working on diabetic retinopathy. Diabetic retinopathy. So take a picture of the back of the eye, and with that, can you figure out is there a diabetic retinopathy or diabetes disease in the eye? Now, these are perhaps the first three, and then this is already a year and a half old. Here are the next 40, okay? And now you can see the timeline on the left. You can see all the different companies in the middle. You can see all the areas of medicine on the right. So it's not just imaging and pathology, but no doubt every field of medicine is going to be impacted by these areas, by AI and machine learning. Whether it's at the molecular level, clinical level, we'll talk about both of those here. There's definitely going to be an impact here. Now, this isn't that new, okay? So these are the first papers going way back to the early 70s. And those of us who are in this field remember this, right? Or some were barely born like me at the time, but we know the history here. AI has been in medicine for a long time, for decades, right? So on the left, there's Ted Shortlift's paper showing how software tools trained with rules, right? I told you about rules. We didn't have a lot of data. Could help a clinician pick the right antibiotic. Because you want to pick the right antibiotic because there could be local resistance to that antibiotic in the bacteria in the hospital. And you can see this is so new. Back then, they literally had the code and the input output literally on the in the paper itself. That could be a crude screenshot if you think about screens back then. On the right is literally a New England Journal of Medicine paper around the same time, Medicine and the Computer, the Promise and Problems of Change. We could literally have that title in the New England Journal of Medicine today. And we still have the problems and promises of change here with medicine and the computer here, right? And what happened, it got so hyped up that there was an AI desert because we didn't have a lot of data. We didn't have a lot of great ways to teach computers. And so the desert lasted, I guess, 20, 25 years, and now it's all back again. And why is it all back? Because we have great hardware from the video gamers of all things, right? That same chip that goes into a video gaming board to help display graphics as fast as possible. Well, it turns out that same chip could be also useful for the computations that are needed in deep learning, right? And deep learning is great for this kind of non-linear learning like images and things like that. We have great software libraries that are free from great universities like Berkeley or companies like Google, and not just free, but even high school students know how to use this stuff. And there are annual meetings and conferences. Very easy to get help on learning how to use these software libraries that are free. And then, of course, in biomedicine, we have these great big data sets to train with. And I'm going to talk about both of these during this talk. The first, obviously, is the molecular revolution, the genomes, genomics, and but it's not just genomics, it's proteins. It's a lot of these kinds of measurements that we're making at a molecular level. But we're not just making more of those measurements, we're making them on more people, more cases. And then the other new big data, again, so old, it's new again, electronic health records. I'm going to end the whole talk with the EHR data side. So I'm going to get molecular first and then get to the clinical side. And we still, of course, have these unsolved questions, right? How do we predict what happens next to a patient? How do we figure out drugs? How do we get to more efficient drug discovery because it takes $1 billion+ as you see to make a new drug? And how do we figure out a whole population? Where is it going next? Obviously, in the COVID times, you know, are we going higher or lower with cases? Modeling is just still so difficult here, and we've got these big unsolved problems that AI can help with. All right, so I'm going to start on the molecular side of things. I got my start way back with molecular data working in Zach Kahane's lab at CHIP. And now 23 years ago, I got my start with these chips on the left there. These gene chips is a molecular tool. You put a sample of tissue or from the operating room or for a biopsy or a molecular lab, and you got to read out of all the genes in the genome. Now for the aficionados here, that's RNA. We all know what RNA is. Now we're dosing patients with it for vaccines, but the RNA are the blueprints on their way to coding for proteins. It's amazing. You put samples on this and you get all of these readout of these measurements. When we went to town 20 years ago with this type of work. But what happened is we got so enamored by making these measurements that already one by one was too difficult. So on the right away, this company Affymetrix came out with a 96 well plate of these chips. And then they had a 384 well plate. And why I use this is to just clearly illustrate that biological data acquisition can also grow exponentially, just like the exponential growth we're used to in the computer world, right? Moore's law, computational power doubling every 18 months. Well, I'm going to argue biological data can double or triple at that type of rate too. Maybe we can argue about the exponents here, but I clearly see exponential growth in biological data. Now, what happened in this world, and look, I'm showing you these gene chips because I still love these things. I carry them in my backpack when I give talks in person, and I could be showing blinking lights and sequencers and things like that. But I love these gene chips. What happened in this field is so many people use these chips, so many that the journals, scientific journals, got overwhelmed with these papers, right? Here's a doctor saying, here's a list that change in that disease, and here are the genes that change in heart failure and cancer. And the journal said, we're getting way too many of these papers. No more papers with these gene chips. Okay, that's it. Unless you're willing to share this data into an international repository so others can double check all the math, and maybe others can use these samples for something else. And that's literally what happened. The precedent for this is now more than 60 years old. GenBank, all right, was sharing sequences with each other, DNA sequences, because nobody wants to retype in this data from the papers. And so the precedent is there in the molecular world and molecular research that if you're going to write these papers and if the field is going to keep growing, you're eventually going to have to share this data with the public. And that's what happened here. Fast forward to August of 2012. You can see I started around 1998, so that's where this graph in the middle was at 0. And in August of 2012, we hit 1 million samples publicly available. Now, not every sample of everything is completely accessible to the public, like citizen scientists. Sometimes you got to prove you're a vetted investigator. Sometimes you got to get approvals from your own home institution, what's called the Institutional Review Board, the IRB, to make sure you're doing legitimate research here. But in general, this is the exponential growth we see in data, especially biomedical data. Now, this is August of 2012. I think we're at 4.5 million samples of this particular kind, RNA measurements, and it just continues to just skyrocket here. So this is the world, right? All of this data is available to all of you, okay? Because what happens in a weird kind of sense is when people get grants, when scientists get grants to make these measurements, they go make these measurements, and then they write a paper about it. And then what do they do next? They go get another grant to go make more measurements. And meanwhile, all of these measurements keep accumulating. Somebody should do something with them, right? So that's the kind of idea here. Just to highlight a couple public data sets that I know and love and want to share with you all. Well, a great one is the Cancer Genome Atlas, okay? You can Google this to find it. And just to give you an example, 84,000 cases of cancer here, okay? Now, those are the patients, 84,000 cases of 68 different kinds of cancer and 22 types of data. So not just the RNA measurements I just showed you, but DNA sequencing, even clinical data, what medications were used, survival or not survive, how long did the patient with cancer survive? There's even some imaging data like mammography or head CAT scans, all of that data available. Now, not every piece of this is completely publicly available because there's identifiable, potentially identifiable data here, but you get a sense how amazing these resources are. And for the most part, most of them just sit there, right? There's a cadre of folks, biomedical informatics faculty that look at this data and try to improve ways to get to it. And some use this data for findings. But in general, most of this data is just sitting there waiting for you, right? For you to have that question and see that the data is here and actually use it to solve your unmet need. That's the idea here. I'm a big fan of this one. I got to put in a plug of this repository called Import because I helped run this one with Northrop Grumman. And this is the next big open data, clinical trials data. Now, I've written a whole bunch of pieces. I know a bunch of people in this audience have as well, to New England Journal and JAMA, that I see a world where clinical trials data is going to be more public as well. We can argue about when and how soon and how much protection do faculty get to write their own papers. But I think we're entering a world because of transparency needs that more of this data is going to be shared. And we share more than 300 immunology ones, including 6 COVID studies at Import. Go sign up for a free account here. And what are we trying to do with all this data? Get to precision medicine, right? So what is precision medicine? Let us remind ourselves from these definitions. Now, 10 years ago, the precision medicine report came out. It's amazing. We should write something about it being 10 years. But the idea was, can we customize the care we deliver to patients using all the measurements from that patient, not just molecular and DNA, but even things like your exercise, your preferences. I like this side effect. I don't want that side effect. And we can't just do it with measurements from you as a patient. We got to have all these measurements on a lot of people and then figure out from there which ones are actually going to make the most sense what is the right care that we got to deliver to you. Oh, I'm losing my camera here. Let's fix this here. All right, I'll just leave it like that. All right, so if you go back to the original precision medicine report, it actually comes down to one figure, okay? And the link is at the bottom here. I love this figure. To get to precision medicine, you see the 3 ovals at the top. We need better diagnostics. We need better treatments. We got to figure out what our health outcomes are. So for the whole rest of this talk, I got about 40 minutes or so, 30 minutes. I'm just going to give you examples of using all this data to get to better diagnostics, get to better treatments, and get to better health outcomes here. That's the whole point of this talk here. All right, let's go on here. All right, let's talk about diagnostics first, right? So what is a diagnostic? There are a lot of medical doctors in the audience here, I can see. A diagnostic helps a medical doctor, a nurse practitioner, a provider to figure out, do you have a disease, right, or condition? You might have imaging diagnostics, right? I love blood tests. I love serum tests or blood tests because it seems like you get a sample of your blood and maybe you can tell if a patient has a diagnostic or not, has a disease or not. So I started this effort with a bunch of folks trying to come up with a new diagnostic for preeclampsia. And we're going to use all this data out there to come up with a diagnostic for preeclampsia. And so just to remind us, what is preeclampsia? This is a really nasty condition to have. I personally know family members with this, where a pregnant woman all of a sudden has shooting high blood pressure, and this leads to an emergency condition where the baby has to be taken out, the mom has to be saved, the baby has to be saved, and then we pediatricians have to keep these babies alive for months, potentially, in the newborn intensive care units, free of head disease, brain injuries and infections and all the rest here. Lots of mortality and morbidity here, super expensive condition, and it affects all people around the world. But the diagnostic for preeclampsia that we mostly use is ancient, okay? It's urine protein. It's not even a specific protein, just urine protein. So I wanted to come up with a better blood test for preeclampsia here, a better test and make it a blood test. And so what do you do? Well, you literally type in preeclampsia at all these websites and see what data do you have on preeclampsia. And we found dozens, hundreds now of data sets on preeclampsia where people have already studied that. And the idea is then you download all those data sets to put them all together. And so the basic methodology here is like a Venn diagram. You remember what a Venn diagram is? They teach this in kindergarten, right? You got these circles and you know, I got this data set and this data set, and that data set. I want to figure out what's in common here, right? Put all the circles together, figure out what are they all seeing? I don't care if one researcher sees it. I only care about if 10 researchers see it or 100 researchers see it. And then you chase those down to figure out, could that measurement be a blood test for a condition like preeclampsia? And so here's an example of one that we found, hemopexin, HPX. We chased it down just for the aficionados here. We had the RNA measurements. We turned this into a protein measurement, an ELISA test. And here you can see in pink that the hemopexin level was higher in the pink, which are the preeclampsia, women with preeclampsia compared to women without preeclampsia who are also pregnant here, right? And you got some p-values and statistics, and it doesn't matter how early in the pregnancy it was. There it is on the right. We had maybe a dozen or so markers here. This was funded by the March of Dimes, and this was during my Stanford days, funded by Spark, which is a seed grant program. Linda Liu did the work, Bruce Ling did the proteomics, Matt Cooper got involved in a moment. We wrote some papers on the bottom right, and what do you do next in Silicon Valley? You start a company on this. And so this became a company called Carmenta, and this is what Matt Cooper did. He was the one-man company here, launched with the whole concept here. And you can see on the bottom left there, secured over $2 million in seed financing here, okay? Now, I love this company. $2 million, by the way, is not a lot to raise in Silicon Valley if you look at any headlines here. But the concept here is that the science continues in the startup company. It is not taboo to talk about companies in biomedical talks, okay? You gotta follow all the rules. You gotta disclose everything, right? I disclosed everything. But the point here is the next scientific experiment now is to do a prospective trial. Okay, we got the grants, we got the papers. What are we gonna do about this, right? We gotta do a prospective multi-center trial, but that trial would be funded by private dollars in the startup companies. So I want you to think about startup companies as a way for the science to continue. Now, I love this one because on the bottom right there, you can see that company has already been acquired. So we went from downloading data off the internet to selling the company in 18 months. 18 months. Inventors happy, investors happy. Of course, we're going to do more of these, and I'm giving away the secrets here, right? Because any one of you on this Zoom can do exactly what we did. In fact, there are so many diseases out there that need diagnostics. We could never step on each other's toes. That's how many diagnostics we need in medicine. Everything I just explained to you is at the links referenced here, right? All of this is open, how we did this. All the articles are there. We literally downloaded the data, and eventually sold the company 18 months later here, right? You too can do this, is the idea. By the way, we have no fancy computers in my lab, no fancy equipment in my lab, right? I mean, we don't even have computers. We're renting them off of Amazon now, right? So the idea is the most capitally efficient way to get to this type of creation here, right? And not ending it with the papers, right? Because again, if you want to change the world, you can't just keep writing papers about it. All right, another diagnostic, right? DNA sequencing, my goodness, is gonna obviously change medicine if it hasn't already. I still remember the days when the Whitehead Institute on the bottom left there was sequencing the genome. I was there in Boston, literally at Boston Children's. And now this is my hand holding a USB stick powered sequencer. This is a nanopore, an Oxford nanopore mini ion. And it's a no brainer when the sequencers are this small. Of course, they're going to be everywhere. Of course, they're going to change medicine. Now, I'm not saying every exam room is going to have a sequencer. I'm not even saying every clinic is going to have a sequencer, but sequencing is going to take over medicine. And it's a big source of data, right? I mean, this is a grand source of data that is already here. Now, I got my start on this one really 10 years ago, actually 10 years ago yesterday, it turns out. Because we got involved, again, my Stanford days, because one of these patients showed up with their genome. In fact, on the left there is Steve Quake. Now, he's a kind of weird patient. He starts sequencing companies on the side, including some in Cambridge, Massachusetts. And he showed up one day because he was the red circle, and his nephew in the blue circle, age 19, so let's say pediatric patient, all of a sudden died one day, sudden death, okay? And so Steve Quake goes to his cardiologist and our friend, Ewan Ashley, and said, well, doctor, am I gonna die of sudden death? And by the way, doctor, here is my entire genome sequence. And with that, he became the first patient to show up with a whole genome sequence. Now, look, you and Ashley could give an hour-long talk on Steve Quake's heart. Suffice it to say, we found in the course of this expedition that Steve Quake has no known mutations associated with sudden death, let's say arrhythmia. He might get a heart attack later in his life, like many in his family have, but at least for arrhythmias, we couldn't find any known mutation. Okay, so that's good. Russ Altman, our good friend and colleague, also got involved because now that you have a genome, what else can you do with it? And he could give an hour-long talk on all the different drugs, pharmacogenomics, that we can now say something about because you have a genome sequence, right? Is the drug going to work? Could there be side effects? What kind of dose could you need? Any of those 150 drugs we could say something about growing all the time. So we got the 150 drugs. Okay, so heart done, drugs done. My lab was stuck with all the rest of medicine, okay? Every other disease condition. Now this was happening, this paper came out, we explained all this in Lancet in 2010, so now almost 11 years ago. And this became one of the most highly cited papers in all of medicine that year. My lab had started with this process and this project back in 2008, because we knew someday patients were going to show up with their genomes, incredible amounts of data, and we were not ready in medicine yet to deal with this. And so how do you get to the data involved here? It turns out that all of these genome differences, right? You have a spelling of this, I got a spelling of this in our DNA. What does that actually mean? It turns out there were so many papers published, but all of those important genomes were locked up in these publications. And someone literally had to read every one of these papers to extract out all the important differences. And so what we did in 2008, I think it was, we started a San Jose high school kid, gave him 50 papers to read, and said, okay, start with these 50 papers, and let's come up with a data model. Let's code every single thing we can get from these papers. What is the actual difference of the polymorphism, the size of the study, what race, what ethnicity, the p-values, the confidence intervals. We wrote everything down. And so we figured, you know, if a high school kid can do this in 50 papers in a summer, how long would it take to just read the entire genetics literature? And that's what we did. So obviously we had to get more of a team to do this. A number of these individuals are actually in India, just reading papers. Literally bioinformatics people, geneticists. So here's one paper. Here's 1,000 papers. And we blew past 20,000 papers we manually curated to make a master list of every gene in the genome, every SNP, every polymorphism, and every different disease. And with that, I believe we still probably have the most master list of every single SNP in the genome and every disease. And with that, then with Steve Quake, we were able to come up with this figure in 2010. And so the idea here is this is how I think doctors and patients are going to talk about their genome. It's not going to be a table of all these different base pairs and DNA. It's going to be this. In the middle here, all the diseases, and the font size goes along with how likely you are to get that disease. Now, it's kind of curious. Hypertension is small there in the middle because there's no genetic risk for hypertension. But by the way, if you end up with obesity or type 2 diabetes, those two conditions can cause hypertension. So you're not off the hook, right? Just because you don't have the genetic risk for this. There are other reasons to get hypertension or any condition. And all along the edge here are all the known environmental or behavioral factors that influence the etiology of those diseases. And we mine the heck out of PubMed to do this. And so, yes, it says you shouldn't, so Steve Quake, you shouldn't smoke, you should watch your alcohol, you should exercise, and you should watch your diet. Probably don't need a genome for those four, I admit. But at the top there, you see pesticides, and Steve Quake has a slightly higher risk for Parkinson's disease. So we would say, if you love to spend time in the garden, stay away from the pesticides. And so to me, this is how we communicate about risk someday, where imagine a figure like this that it's constantly changing, right? As you get older, you're not going to get those pediatric conditions. You're going to get some of the geriatric conditions. And the risks are going to constantly change depending on what you're doing, what your behaviors are, maybe from your wearable and all the rest. I think this is going to be an interesting way to talk about risk. And so this is the figure from the paper almost 11 years ago that we were promoting. Of course, genomics has totally taken off from those days. We use it for cancer, we use it for neurological conditions, we use it for so many different things, genome sequencing. But what did we do in Silicon Valley? We started a company on this. This became a company called Personalis. It was literally incorporated 10 years ago yesterday, launched with the sequencing of the Million Veterans Program. You might have heard of that process and the project in Boston there. Raised more than $76 million from private investors, IPO'd a year and a half ago, and today is a $1.5 billion market cap company. And again, the science continues in the startup companies, right? They're still doing science, still doing research and development. It doesn't end when you start the company. But to put it a different way, do you know how hard it is to get a $1.5 billion NIH grant? You know that's impossible, right? There's a certain level of science that is just going to get done in the company now, right? We take it as far as we can in academia, and it has to go further, right? That's the whole point here. All right, let's keep going. Drug development, okay? So we're done with diagnostics. Now we need therapeutics. How much does it cost to make a new drug? A ridiculous amount. Matthew Herper does this every couple of years, at least he was when he was at Forbes. This is the 2013 numbers. The top 12 companies spend between $3.6 and $11.7 billion per drug. Probably not sustainable. Now, look, I'm not going to pass judgment on the pharma industry here, right? It costs a lot of money, and sometimes it's pretty high risk because you got to fold in all the failures, right? It's a hard problem. And I'm not saying I got the world's solution to this problem because it's a hard problem. But boy, can't we use all this public data to at least make a difference here? And the idea here is we got all these folks depositing data on diseases and a few depositing data on the drug side of things, especially including the Broad Institute over there, right? Why can't we start to combine these kind of data sets together? And not just find new drugs, but maybe a shortcut is to find new uses for old drugs. And so my lab did a lot of these. We got, 15, 20 papers, some still coming out, where we did a whole bunch of these drug repositioning, data-driven drug repositioning studies here. Here's one of the more recent ones. This is about 3 or 4 years ago. Bin Chen and a whole bunch of folks at UCSF and Stanford did the validation on this one. This is for a condition called hepatocellular carcinoma, or liver cancer. Now, liver cancer affects, I think, 350,000 people a year, but not that many people in the United States. And I'm going to argue because of that, we basically have no drug companies working on liver cancer, okay? This is an India-China problem, where because of viruses and other environmental conditions, they end up with liver cancer much more than we do. And there are no drugs for this. So we turned our computational crank, and here's the answer we got. Nicolosamide showed up at the top of the list. So this is a mouse liver. What you're looking at are pictures of mice liver. I'm before lunch, you're before dinner, so apologies for that. But you're looking at mouse liver there, infused with human liver cancer, okay? And on the left, there's nothing being done to that mouse, so that's how nasty it looks. Nicolosamide, wait a minute, you said this works. That doesn't look that much better. Yeah, I admit you're right. NEN, though, is nicolosamide ethylolamide. So it's a particular salt of the nicolosamide that this mouse is eating, okay? You don't even need an IV, you just eat this drug, okay? And you can see it looks a little bit better there, a lot better. Here's glowing cancer. You can see NEN after treatment. There's some mice that's way smaller there. And there is one existing drug for liver cancer. It's called sorafenib by a company called Onyx. And sorafenib is actually easy enough to spit out. And so here you can see how this drug works with sorafenib. So you got nothing done to the mice at the top, NEN by itself in the second row. Sorafenib is the third row. You can see, again, the cancers are smaller. But when you combine the two, wow, it's even better than either one of the drugs separately. So that means there might be synergy going on here, right? These two drugs might be working in different ways. And because of that, maybe if you're going to run a human trial, you're going to want to dose them with both. By the way, nicolosamide is a well-known drug. We use it to treat worms. The infectious disease folks among you might remember that. I don't even know if it's still approved or not. We use it in veterinary medicine as well, but it's used to treat worms. I mean, worms in your GI tract. So this drug is also useful for cancer. Now, we did a whole bunch of these. Of course, there's many people doing this now, okay? There's so many now, I'm kind of exiting this field. I'm not doing any more of these. So we still have a whole bunch of papers that are coming out, but we're not doing any more of these. But what do we do in Silicon Valley? We started a company on this. So this became a company called Numedi, raised, I don't know, $6+ million, a whole bunch of pharma deals. And now they're working on a drug for idiopathic pulmonary fibrosis. Nasty condition where the lungs get fibrosed. There are 2 existing drugs for this. Neither is that good. So they're working on, they got a great candidate for a new drug for IPF. And you can see, this is what you do. You start with the unmet need, the data is available, get the work going in academia, but follow all the rules, spin it out, and make sure it continues. Otherwise, patients are just not going to get help here. All right, so in the last few minutes, where are we going next here, right? I'm going to now switch out of the molecular world and talk about the other big data, electronic health record data. Oh my goodness, this data is so much more expensive than that molecular stuff. In fact, we're spending billions of dollars collecting this data. Here are just 3 great examples. Press releases illustrating how much it costs to put in these EHR systems. Sutter, which is a medical system around here, us in California, spent $1 billion on their EHR system. Partners, which is of course Mass General and Brigham, and the affiliates spent $1.2 billion on their system. And the Kaiser Permanente spent $4 billion on their system, right? Now, amazingly, they all bought the same system, Epic. So the price tag can hit $1 to $4 billion for software. Of course, it's not just software. It's workflow redesign, it's training, it's all of that. But man, is this expensive. Now, the kind of punchline I want to make sure you all get is that given the billions we spent on this, on putting in these systems, it will be a national tragedy if we don't use this data to improve the practice of medicine. Of course, we have to do it safely and responsibly. But what a tragedy it will be if we've spent hundreds of billions of dollars here and we never use this to improve the practice of medicine. That's the narrative I want to make sure you get. I call this the most expensive data in America now, okay? We pay doctors to type this stuff in, all right? It'll be a tragedy if we don't use it. And so that's the mantra moving forward. Of course, safely, responsibly, and we'll talk about that. So with that, let me reintroduce the University of California to you. And why did I move to UC after 10 years of being at Stanford? University of California is incredible. 10 campuses, 3 national labs, including Lawrence Livermore, which I think has the #1 supercomputer in the country at least, right? We help staff that national lab. 200,000 employees and 250,000 students per year. We're actually one of the larger employers in the United States. But like most employers in universities, we're still trying to figure out what to do with our employees and students given COVID, of course. 20 health professional schools. So that's 6 medical schools, but we have nursing, pharmacy, dental, veterinary, public health schools, all the rest as well. We train half the medical students and residents of California. 2 billion in NIH funding, 13 billion a year seeing patients, 5,000 doctors get a paycheck from us, but 100,000 doctors write orders on our patients, 12,000 nurses, and we're not just random centers. UCSF and UCLA are both in the US News top 10. Of course, we would only mention that if we're in the top 10, otherwise we don't care so much about that metric. And we have 5 NCI comprehensive cancer centers and 5 NIH CTSAs, Clinical Translational Science Awards, which is marks of excellence from the NIH perspective. We also have these secret superpowers, which are so secret, sometimes we even forget we have them. IRB reliance. If any one of our UC campuses approves an IRB, the others will rely on that one campus and automatically approve it, more or less. So it's days to weeks instead of weeks to months, let's put it that way. And centralized contracting. So if a pharma company or any company contracts with the office of the president, then all of us agree to the terms, we don't renegotiate. So our ability to scale is crazy, okay? We can scale like crazy. And what do is we announced 4 years ago, now maybe, a little bit more than 4 years ago, is that the entire University of California is going to now form a single ACO, or accountable care organization, to represent our ability to take care of patients to those that need it. For example, employers. Now, ACOs come out of the Obama administration, accountable care. The idea here is that we as healthcare institutions are going to take on risk. That means you're going to pay us per member per month, right? Certain amount of money. And no matter what happens to that patient, we own it, okay? We own that problem for that patient. So you're contracted to take care of those patients per member per month. That's accountable care. But so UCSF has ACOs and UCLA has ACOs, but now we're talking about aspirationally, this is a 10-year plan with Optum and United Health, okay? Again, we got to learn how to take on risk. Might as well learn from the best. But the idea here is to now really represent the entire University of California as one. That would kind of be the equivalent of Harvard doing this, which would never happen, okay? All the medical entities in the Harvard ecosystem, right? Never mind just in Boston, right? The competitors right across the street from each other, right? So Harvard can do that, UC can do this here, because we're far apart enough that we don't compete with each other for patients. That's the beauty here. So can we leverage our scale to actually do this? But the first question that comes up immediately is, whose style of medicine are we going to practice, right? So like UCSF might do it this way, and UCLA might do it that way. Which is the right way to do it? What's the UC way going to be? And to figure that out, all of a sudden you got to put all the data in one place to figure out the right UC way to do it. And that is one major reason why we came up with a central database for all of this work. Of course, we had academic need, grants, papers that wanted data to come together across UC, and we had a lot of efforts made using I2B2 and UC Rex, but we also had now the operational reason to do this, right? We had business reasons to put all this data together, and that, in my view, is what really made this happen in the end. So of course, I wouldn't be an IT guy if I didn't know how to make boxes point to boxes. So here's the one slide like that. You can see the 6 logos at the bottom. So look, Riverside is tiny, brand new medical school. They don't have a hospital system there yet. We have partners there, affiliates. So they're tiny. The other 5, San Diego, Irvine, LA, San Francisco, Davis, are massive medical enterprises. Okay, they're huge medical enterprises. And all that data now comes and goes into the central data warehouse. And we were happy with this happening every month. We've actually sped up to every 2 weeks. And with COVID, we do this every day now. So every night, all COVID data comes centrally, so we can report to many different folks. I'm not going to otherwise talk about COVID here. All right, so these are our latest numbers. The top line we love to start with is we've taken care of 15 million patients in the last 15 years. It's kind of a round number because it's 5% of the entire US population has gotten care from the University of California. But if you talk about when we actually installed Epic, which is the modern, Epic and modern, same sentence, I get it. But when we installed Epic in January of 2012, moving forward, we've seen just shy of 7 million patients. I'm not even counting affiliates. These are just main clinics, main hospitals on our main campuses. 6.7 million patients, 200 million encounters, 500 million procedures, 750 million medication orders, my favorite, 2 billion blood tests and vital signs. And as I like to say, we have everything from Tylenol to CAR T cells. Tylenol is probably the cheapest thing we can do to our patients, give them a pill of acetaminophen. CAR T cells, of course, when you take out someone's blood cells and train them to fight their own cancer, is probably the most expensive thing we can do to a patient. We have everything in between those two. We have state regulatory data, California OSHPA data, text elements, death index, when patients die in California, and we come up with all these dashboards, which I'll show you a couple examples here. First of all, I want to make sure everyone gets this. I'm going to show you all the reasons we benefit from having this data, but all UC patients can download their own data using a federal standard called FHIR. I forget what FHIR stands for, so Ken's going to kill me because of that. And it's not just available in FHIR, it's available in Apple Health and Health app, including UC San Diego was a launch partner with Apple on this one. But if you literally go to Apple, if you happen to have iPhone, you go to health data, health records, you type in UC, you're going to see all of our campuses there. Now, we know not everyone has an iPhone. We know people have Android. UCSF has partnered with Common Health to get the Android version of all this built as well. So we got to make sure this is equitable. Now, it's another exercise left to the listener here. What are we all going to do with these records? Because not a lot of people have used this yet. I got some ideas, but so far, we keep giving more and more data to patients in standards. They haven't done much with this yet. And so we should think about why and what we need to do next step. But anyway, patients are going to benefit. At least we're putting this data out there. All right, so this is what it looks like. This is reasonably recent, about 6 million patients in this part of the country. Yeah, we love Tableau. These are our standard kind of colors. You can see them again and again. And this is a map of California on the left there. So you see San Francisco and Davis in the northern part. You got LA, Irvine, and San Diego at the southern part. There's a tiny red dot you can't really see for Riverside. They only have a couple thousand patients. And then you see Las Vegas there. I don't know if you can see my mouse. Because of course, if you're sick in Las Vegas, you're going to come to UC. Hawaiian Islands are covered with our colors, for example. Of course, age, race, ethnicity, gender. Of course, we have all of that. The diversity of the world is in California and is treated by the University of California. By the way, we have every telehealth encounter as well. This is kind of fun. We've now blown past, I think, 1.5 million telehealth encounters in our database, okay? Central database, 1.5 million telehealth encounters. Can you imagine AIing the heck out of this, right? So if we can do this remotely, what exactly did we do? Did we prescribe a med? Did we do a lab test? Who's come back for more telemedicine? These are the papers we're writing now, guys, okay? We've blown past 1 million of them. We're almost close to 2 million. This is public data. You can see telehealth has just obviously took off in March last year and continues to go and grow, okay? It's not going away. Telemedicine's not going away. We've improved a lot of different lives in the University of California, patients including, but we've also saved the financial side. Central tools to improve quality of care, decreasing expenses, central management of primary care patients. I'm going to show you a couple examples of these. Here's an example of quality reporting. Now, can you imagine this happening in Boston? Let me put it this way. I could never imagine this happening in Boston. We got here just 3 academic medical centers all using the same quality measurement tool, but not just quality measurement. This is quality prognostics. So, for example, we have to report quality all the time to payers, to Sacramento for Medi-Cal. And here, an example, these are names of doctors, not patients. So this is de-identified. And this is old data now. But you get an example that we don't want to just say 3 months ago, patient should have been counseled to stop smoking or colonoscopy for colon cancer. Here are the patients literally coming in tomorrow at 3:30. This patient is missing 3 quality measures. Somebody go and make sure they've stopped smoking, make sure they're not on opioids or addicted to opioids, and document it in Epic. And it can be documented across, we have 7, 8 different instances of Epic. It all comes down into the central data warehouse, so we can constantly do this. So we can divide and conquer because we're all using the same data model, OMOP. Our operational teams are using the same database as the research teams. I want to make sure people get that because it's remarkable. When the operations and quality improvement team are using the same database as the researchers, then the same dollars get the same thing to happen, right? You don't have to duplicate or triplicate work here. That's why this all happens. Let's skip forward a little bit. Another killer thing that I've learned, okay, is not just to take care of patients, but to take care of our own. So it turns out, and there are Boston hospitals that do this. I remember how I said we have 200,000 employees. Well, maybe a third of our employees and their family members get care from us. So they don't just work with us. They're not just employees, but they also get their care from us, right? Now, you have to realize that that's kind of rare. I've visited many academic medical centers where the faculty there would never be dead getting their care at their own hospital, right? That's really sad if you think about it. Of course, our employees are proud of getting care from us. In fact, they like the care. But then if you think through the incentive arrows, every dollar we waste on our own patient care, right? I'm talking about waste here, is actually equivalent to burning money on the front lawn, right? Because now the care we deliver to our own employees is a cost center to us, not a revenue center. And I still am going to believe this is the way you're going to improve care and improve the technology development by showing how much you can save in money by doing this kind of work, not to the researchers, but to the CEOs and the C-suite. What you're looking at here is a graph that's going down. This is a long story, but I'll make it all simple about diabetes. In fact, there's this drug we use for diabetes called metformin. It's so old, right? There's generic forms of metformin available. But we noticed our own employees in our self-funded health plan, right? We pay the cost of our healthcare for our employees. We noticed a bunch of our employees were getting prescribed brand name metformin instead of generic metformin. That's kind of strange. Why are they getting prescribed brand name instead of generic? The generic's available. But guess what? It was our doctors writing those prescriptions to our employees. So you think about it, you realize you have some control here, right? So we call all the patients and you can get some of them to switch, but everyone loves the color of their pill. You can call the doctors and all of a sudden they realize you're looking at this. So now they're going to start to change their behavior. That's the Hawthorne effect. And you make it harder to start new patients on brand name metformin. And just the one drug, I think we're close to $2 million saved on just the one drug so far for the University of California. You do enough of these, this pays for the whole database, okay? The whole thing is paid for with just a couple of these. Never mind everything else we're doing, right? And so in this magical world now, the research use is actually marginal, if not free, because the operational folks, the health systems love this enough and they're saving money that it actually pays for itself here. So that's the kind of example I'm trying to get to you, that this is the new world that we can actually do this kind of work and save money in this way. So I'm going to end. Let me just fast forward here to the last slide here by thanking a whole bunch of folks. And we can go into some more in terms of the questioning here. But I can already see the Q&A bubble has been going like crazy. So let me just share. I have to thank a whole bunch of collaborators who have helped me over the last few years at NIH, Stanford, and UC and UCSF. I have to thank the team that actually puts this data together. Incredible team on the clinical side to make all this work happen. And obviously thank UCSF and Priscilla Chan, Mark Zuckerberg, and the Baker Family Foundation for their endowment support. My lab has been blessed with 21 NIH grants from these 11 institutes of NIH. The ones in red give me more money. The ones in orange give me less money. I still love them. Whole bunch of disease foundations. Thank my admin and tech staff wherever I go. I always thank my family and those who've supported me. And of course, Zach, who has been my mentor and friend since I started as a researcher. So let me stop it there and see if there's any questions.
Atul, a spectacular talk as always. And there are, as you know, questions bubbling up in the chat and the Q&A. Let me start by asking you a couple questions of my own. So my guess is when you arrived out in California, that the 5 different hospital systems that you oversee didn't just come to you and say, how can we give you control over all of our data and data science? And so can you talk a little bit about the sociology, which is an incredible part of this magic trick that you've shown us, that could not have been entirely easy?
Absolutely. I think you're breaking up a little bit, Ken, but I got your question there. So in fact, they did, okay? That was literally the reason I moved, is because they had just really created this enterprise called UC Health, this umbrella. And we had already seen the clinical need, the operational need, and the research need all wanting a central database. So my role coming in was really to build this data warehouse. Now, how did it happen? Step one is you can't step on any, right? So each campus, every health system has their own academic team, their own analytic team. So we don't want to step on their toes. So we were answering questions really that no one else was going to have the ability to do. Second is save money right away, okay? Or really add value right away. So we picked a pilot first. The pilot was to look at one quality measure, statin compliance. Some of you might know primary care patients should go on statins if the lipids are abnormal. And so what we did is we created a simple dashboard using central data where we could see how many patients should be on statins and how many actually were on statins. It's kind of interesting, okay? So we had the 5 main campuses for us, and the national average is somewhere around 30%. And then we said we had 5 bars, and the first was 50%, 50%, 50%, 50%, 17%, okay? So one was much worse than the other 4, and that one was actually worse than the national average. And then we realized, you know what? We got to look at all these quality measures and fix them before we're forced to report them to everyone else, right? That's the whole point. It isn't to always be a rear view mirror. We should be fixing these problems with quality, right? And so that's how we got into this. We started to right away show divide and conquer. We can implement quality measures within days to weeks. It doesn't take us months to do that when a new payer comes in with quality measures. Once all of that was set, then we opened this back up to researchers, because researcher dollar flow is much smaller than operation clinical dollar flow, right? Once it's essentially paid for by the clinical dollars, the research use is just marginal to free, and you're enabling researchers like crazy. But it really needed us to realize we can do better business together by acting together, right? So another example, UCLA is buying this drug. UCSF is buying this drug. Why don't we put one purchase order in and get a better price for this drug, right? Leveraging scale for value. We literally call it LSFV, but it's enabled when you have data in one place, right? But the important thing is we're geographically far apart, that we don't compete. And especially in Boston, how local healthcare is, especially competition.
Outstanding. And so the sociology of it revolved around value in the healthcare sense of the word value.
Absolutely.
So you leverage that, and you also, unlike many organizations that are seeking value, turned it also into an academic engine.
That's right. I think exactly. The academic engine is much easier to launch and relaunch after you built the thing, right? And so once we built it, remember, it's operational, so it's fully identifiable. Then we can then do the HIPAA de-identified version, and then limited data sets in between. So we literally have bookends, fully identifiable, completely de-identified, everything in between. And then any of those could be available for research. In fact, now our de-identified central data warehouse is so de-identified and has already been ruled on by all UC IRB directors as non-human subjects research, okay? So any UC researcher, including grad student and postdoc and faculty, can get access without further IRB approval, okay? And it literally lives on the Azure cloud in something called Databricks. It's one of these cloud data environments. And literally, you can build a machine learning tool that's learning from 130 million ambulatory visits. Now, one of the questions I saw in the Q&A, is this data going to be publicly available? No. In fact, I did a bait and switch on you. I start with public data, then I talk about clinical data, right? We're far away from having clinical data available publicly, okay? But we've opened this data up to all UC researchers. You want the biggest centralized database, I'm going to argue, come work for us. You get it right off the cloud right away in a way that no one else has right now. All the NOMA standards.
Very nice. Let me go to a few questions from the chat. And since you were just really talking about HIPAA de-identification and public-private, let me go to a question that you can answer in a directed way. Isn't patient privacy a problem for you?
Yeah, so first of all, of course, I keep using the phrase safe and respectful use, okay? First and foremost, you gotta make sure everything we're doing is legitimate, and that means we turn down many collaborations because we don't think this is safe or respectful use for data. We have a chief health data officer, Cora Hahn, who came to us from the Federal Trade Commission. I'm gonna argue she's the very best at this, having really brought many Silicon Valley companies to task for inappropriate use of health data. So we have a justice-based model that we've been using, making sure patients know what we're doing. And it's de-identified. I mean, so now de-identified doesn't mean we make it open though, right? So we're still very concerned when the data is de-identified to make sure people can't re-identify, especially tech companies know how to do that. We want to make sure we're protecting from that. Now the text elements, we've gone crazy, as you can see from some of our press and publications, on how to de-identify text documents. And I don't mean just writing papers. I mean, I want to sleep at night and not wake up in handcuffs de-identified. We believe we're there. We believe we're probably the best in the country at this. We have a tool called Filter, and we've already applied it to all 105 million UCSF notes. Our best estimate now is we're probably at 600 million notes UC-wide. We're applying it to that next. Of course, we're doing concept identification, all that too, negation detection. But we want to get to safely de-identified notes in a way that is still usable. We're also doing this for images as well. So you've seen a couple of papers from us. So that is the repository we're building to do this safely. But in all aspects, not hiding this from patients. Patients got to know. And we're doing this to improve the care we deliver to patients, hands down. Otherwise, if patients aren't going to benefit, we're really not sure why we're going to do it. Okay. And that includes collaborations as well.
Outstanding. Great answer. I have another question for you. And that is from Adam Culbertson, who's actually from the SMART team, formerly at ONC and other places, now at Humana. And he asked a question that's near and dear to his heart. What method do you use for record linkage?
Great question. So first of all, it's dumb luck. Most patients get their care at one UC and not the others. Okay, that would be more of an issue in Boston, let's say. But we can also use some kind of soft linking, so date of birth and names. We don't have a formal EMPI, so there's commercial products you can buy for that. Our campuses already use EMPIs for their affiliates, but centrally we'll use crude kind of name matching. We have ones that we know are match or direct matches. We have some software matches there. But that we're not innovating that field. That's stuff that we're borrowing from established kind of papers and things like that.
Great.
If it wasn't clear too, we're using OMOP as a backend. I keep saying it just to make sure. Man, OMOP again, it's one of these things that's so old, it's new again. So we're all over OMOP. Go ahead.
Yeah,
I can stick around for a couple of minutes too, but go ahead.
There's a question in there about how do you compare OMOP to the I think they meant Epic backend, and so a brief word on that.
Yeah, so it's incomparable. Epic clearly captures much more than we can represent in OMOP, right? It takes time, money, and effort to convert things into OMOP, right? So, for example, most of our OMOP harmonization work had been around primary care and population health management, diabetes, what they call population health, that area. And so we had all those drugs harmonized and lab tests harmonized and all that. That's great. And then COVID happened. And all of a sudden we had to harmonize ECMO settings, right? That's when the machine is completely taking over your heart and lungs. But UCLA had already figured out the SNOMED codes for all the ECMO settings. The other 4 campuses literally took what UCLA did and figured out where their Epic settings were. So of course, Epic has much more than what we can represent in OMOP, right? You can't boil the entire ocean, so you pick corners of the ocean first to boil. All of a sudden, we switch to the intensive care unit side and harmonize all ICU things for COVID, right? So now we got, we can figure out the ECMO setting across the whole UC. I noticed one of the questions on pediatrics. We've done a lot on pediatrics. Of the whole set of the 7 million or so, I think 1.1 million of them are kids. Okay, I'm obviously a pediatrician. So we've got 1 million kids in this database, right? All sorts of things we do with kids, right? So care delivered. So I'm going to argue it's just incredible data we have here. And again, for the machine learning aficionados, right? Think about it. You're used to machine learning where you can leave one case out validation, right? I want to train my machine learning, but I want to leave one case out so that I can test on that one. Imagine we can do leave one academic medical center out cross-validation, right? That's how many academic medical centers we have in a central database. Now, the questioners have asked about N3C, the National COVID Consortium. We love it. We'll be participating. It's there. It's good. It's COVID. Already too many negatives. We're going to cut here and paste there. So we're watching that carefully, and we think it's great. But we have our own UC one as well, but we have our notes in there and stuff like that too. So your own health systems are always going to have more data than you're going to give to others, I think.
Yeah. And we have a question about federation. And I'll paraphrase the question from Brett Johnson. Are you going to federate any of your DID data with systems outside the UC system?
We could, but it's going to have to be of interest to us and our patients. Okay. Now, we had a federated system for the longest time, and Ken knows UC was, I think, the customer number 2 of I2V2, right? We had a system called UC Rex, and that's also what helped with the sociology. Lucilla Machado, Doug Bell. I mean, the teams were incredible. Even Lisa Dom back then were really making sure that these teams were constantly talking to each other to make sure that when a drug came out, we had the harmonization happening, monthly discussions that have continued for a decade, right? But federation is at the weakest link, right? It's as strong as the weakest link. Every time I would query something in I2B2, one of our sites was down. You could do this now. You could go to the CTSA Act, the ACT, and every time you query it, every couple of days, it'll be a different answer. Some site is on or offline. That's not a way to do it, just to be blunt. If you can get decentralized, it's worth getting decentralized, right? That's a no-brainer. If you can't, then all you have is federate, right? I get it. I totally get it. But centralized enables you to do things that federate just is going to have a hard time doing. The reliability isn't there. These projects are just not as they're not as prioritized on the campuses. That's the problem.
Here's a question comparing what you're doing on the West Coast to what might be happening in public system, for example, the NHS. What are some of the differences, do you think, in the challenges that you're facing in a public system versus an NHS style system?
Yeah, so I'm going to argue we're also a public system, public-private, let's say.
Yeah,
the UCs are a state university.
Absolutely.
You know, so, but to be really kind of coy about it for a second, we're public when we say we need to be public, we're private when we say we need to be private too. So we're kind of in between. The NHS is there, but remember, the NHS has a lot of data integrated, but not 100% either. And they've spent billions with Accenture and others trying to get better data in a central data repository. It's hard there too. It's hard there too. So, but my take is that if you have this data, it's your responsibility to use it. And so I'm going to challenge the world as we have this data, that we should be doing more with this data. And so one example of what we should be doing more of is constantly looking at our own data, figure out what's working in medicine, what's not working, what is too much, and what is missing. Okay, and we really should be the ones to say, you know what, this drug, this expensive drug is not working any better than this cheaper drug. Okay, we should be the ones to say that, with or without a grant. Okay, it shouldn't need a grant for us to do that kind of questioning, right? We should do that questioning constantly to improve the care we're delivering. And yes, NHS has this, and Denmark has this, and a lot of these countries have that. I don't see them asking those questions, right? Now it's getting there. Real world evidence is getting there. Everyone's getting there. But it's about the responsibility to use this data to really say, okay, we're not going to do X, Y, and Z anymore, even if we make money doing it as a health system. That's going to be the hard part. It's about responsibility, not just technology.
Atul, amazing talk, great answers to questions. There's more questions, but I'm just getting a sense that it's 5:09. The sun is starting to go down here. I think this is the perfect time to end on a very, very high note. And so I'm going to thank you. I'm going to share my screen one more time and just finish up here. Hang on for a second. Upcoming talks are next week. And Atul, you might be interested in this one. Lawrence Lessig, founder of Creative Commons, founder of the Electronic Frontier Foundation, Harvard Law professor. Then in April, Enrico Coyera, one of the foundational thinkers in biomedical informatics for decades, and director of the Center for Health Informatics in the Australian Institute of Health Innovation. Coming up, and we're scheduling this for a little later in the spring, Michael Creamer, Nobel Prize laureate in economics 2019, who has advocated and implemented randomized clinical trials, something that there was a question for Atul about in the clinical trials. We didn't get to it today, but that's another interesting opportunity when you have large amounts of data that's organized in a healthcare system. And once she comes back from her maternity leave, Chrissy Farr, former CNBC health tech reporter extraordinaire, now a health tech investor at OMERS Ventures. So You can find us at chip.org. If you registered for this event, which by definition you did, you'll be notified if you would like to be of future talks in this series. Follow us and follow Atul. And thank you very much for attending. And Atul, again, so great to see you. We should have never let you go.
Thank you very much. You're too kind. And for those who have questions, just shoot me an email or DM me on Twitter. I'll get to them when I can. Thank you.
Bye now.